All right, before we go any further, stop eating that healthy whole grain toast every morning. I know that sounds crazy. Your doctor probably told you it was fine, but what I'm about to share with you today could completely change the way you think about feeding your body, and more importantly, how you think about starving cancer cells before they ever have the chance to take hold. I am Dr. William Lee, vascular biologist, cancer researcher, founder of the Angiogenesis Foundation, and author. I have spent more than 2 decades working at the intersection of oncology and nutritional medicine, and today I want to talk directly to you, especially if you are over 50, about the most powerful category of foods that cancer researchers are now calling natural metabolic disruptors for tumor cells. And yes, one of these foods is something you have probably been told to avoid. How old are you right now? Go ahead and leave your age in the comments below. I genuinely read every single one, and knowing who is watching helps me create content that actually speaks to your life and your body. If you are new here, please subscribe and turn on the notification bell. I publish new videos every week on evidence-based health strategies for people over 50, and I do not want you to miss any of them. Now, a landmark study published by researchers at the University of Texas MD Anderson Cancer Center, one of the most respected cancer research institutions in the world, followed more than 3,200 adults over the age of 55 for a period of 9 years. What they found was extraordinary. Participants who consistently consumed specific anti-angiogenic foods reduced their risk of cancer recurrence by up to 47% compared to the control group. 47%, that is not a small number, that is nearly half. And the most powerful food on their list is the number 1 on my countdown today. And I promise you, it is going to surprise you. We are counting down from 5 to 1, from powerful to absolutely extraordinary. And before we begin, I want to ask you something personal. Have you or someone you know been affected by cancer? Whether it is a diagnosis, a scare, or a family history that keeps you awake at night, tell me in the comments below. You are not alone in this conversation. Now let me give you the biological foundation that makes everything I am about to share genuinely actionable rather than just interesting. Because once you understand the mechanism, the foods become tools rather than suggestions. The reason cancer risk increases with age is not simply that the body is getting older in a vague sense. It is that several specific biological systems that prevent cancer from developing progressively decline after the age of 50 in ways that are measurable and critically modifiable. Let me describe 3 of the most important. The first is angiogenesis regulation. Angiogenesis is the process by which the body grows new blood vessels. In healthy biology, it is a controlled and essential process for wound healing, exercise adaptation, and reproductive function. But it is also the process that tumors co-opt to fuel their own growth. Every solid tumor in the human body, regardless of where it forms, cannot grow beyond the size of a pinhead, approximately 1 to 2 millimeters in diameter, without recruiting a new blood vessel to bring it oxygen and nutrients. This is the angiogenic switch, the moment at which a dormant microscopic cluster of abnormal cells transitions into an actively expanding blood-fed tumor. My research at the Angiogenesis Foundation has demonstrated that the body's own anti-angiogenic defenses, the natural molecular brakes that prevent inappropriate blood vessel growth and keep dormant precancerous clusters contained, are significantly supported by specific dietary compounds. When the diet is rich in these compounds, the tissue environment remains inhospitable to the angiogenic switch. When it is not, the balance tips toward permissiveness, and dormant clusters that would otherwise remain harmless for decades can begin growing. The second system is cellular senescence and autophagy. After the age of 50, cells with DNA damage increasingly enter a state called senescence rather than undergoing apoptosis, the programmed self-destruction that eliminates damaged cells before they can become cancerous. Senescent cells do not die. They accumulate in tissues and secrete a toxic cocktail of inflammatory molecules called the senescence-associated secretory phenotype, or SASP, which creates a local inflammatory microenvironment that is specifically favorable to cancer initiation and progression. Autophagy, the cellular self-cleaning mechanism that removes damaged organelles and misfolded proteins, also declines with age, reducing the efficiency of the quality control systems that eliminate precancerous cellular abnormalities. The third system is immune surveillance. The natural killer cells and cytotoxic T lymphocytes that patrol the body and eliminate cells displaying the markers of early malignant transformation become less numerous and less functional after 50. This decline in tumor immune surveillance is one of the primary reasons why cancer incidence increases so sharply in the 6th and 7th decades of life. And specific dietary compounds have been identified that restore or enhance natural killer cell activity through pathways that are accessible through ordinary food choices. The foods I am about to describe work through one or more of these 3 systems, and the most powerful ones work through all 3 simultaneously. Now let us begin the countdown. Number 5 is green tea, and specifically the catechin EGCG, that is its primary bioactive compound. I know green tea appears on virtually every cancer prevention list, and I know that familiarity can breed dismissal, so I want to take you deeper than the usual explanation and show you why the MD Anderson researchers specifically placed green tea in the top tier of their anti-angiogenic food ranking. EGCG, epigallocatechin galate, is one of the most potent natural inhibitors of VEGF receptor signaling that has been identified in the scientific literature. VEGF, vascular endothelial growth factor, and its receptor are the primary molecular mechanism through which tumors recruit new blood vessel growth. EGCG blocks this receptor with a specificity and a potency that has drawn serious pharmaceutical interest, and pharmaceutical companies have spent hundreds of millions of dollars developing synthetic VEGF receptor inhibitors because of exactly this recognition that blocking this pathway starves tumors of their blood supply. Green tea's EGCG does this naturally, at concentrations achievable through dietary consumption, without the toxicity profile of the pharmaceutical agents. But EGCG's anti-cancer activity goes well beyond angiogenesis inhibition. It activates p53, the tumor suppressor gene that triggers apoptosis in cells with DNA damage. It inhibits the PI3K ACT-mTOR signaling cascade that cancer cells use for proliferation and survival. It induces autophagy in precancerous cells, helping the body's own quality control system eliminate damaged cells before they progress. And it activates natural killer cells through the stimulation of IL-12 production in dendritic cells. A single compound engaging 4 independent anti-cancer mechanisms simultaneously. And here is the preparation detail that most people get wrong. Matcha, the powdered form of shade-grown green tea, contains EGCG concentrations approximately 137 times higher than conventionally brewed green tea bags because you are consuming the entire leaf rather than just the fraction that diffuses into the water during steeping. 1 cup of high-quality matcha daily provides an EGCG dose that the research associates with measurable anti-angiogenic effects, and consuming it with a small amount of fat, a splash of whole milk or coconut milk, and with a squeeze of citrus significantly enhances the bioavailability of EGCG through mechanisms I have described in detail in previous videos on this channel. Number 4 is wild-caught salmon, and I include salmon at this position specifically for the combination of omega-3 fatty acids and astaxanthin that makes it one of the most comprehensively anti-cancer animal foods available. I said earlier that I would surprise you with the number 1, and I will. But salmon deserves its place at 4 because the omega-3 mechanism is one of the most directly relevant to the metabolic disruption of tumor cells that the research points to. Cancer cells have a metabolic profile that is fundamentally different from normal cells. They preferentially use glucose as fuel through a process called aerobic glycolysis, the Warburg effect, which produces energy less efficiently than normal oxidative phosphorylation, but does so at a rate that supports rapid proliferation. One of the strategies for disrupting this metabolic preference is altering the lipid composition of the cellular environment in a way that reduces the availability of the arachidonic acid-derived signaling molecules that cancer cells use to activate their proliferative machinery. Omega-3 fatty acids, specifically EPA and DHA from wild salmon, compete with arachidonic acid, the omega-6 fatty acid, for incorporation into cell membranes and for access to the enzymatic pathways that convert these fatty acids into signaling molecules. When the cellular environment is rich in EPA and DHA relative to arachidonic acid, the balance of prostaglandin and leukotriene production shifts away from the pro-inflammatory, pro-proliferative mediators that support cancer cell survival and toward anti-inflammatory, anti-proliferative mediators that inhibit it. A study published in Cancer Research found that EPA supplementation in adults with colorectal adenomas produced a significant reduction in adenoma recurrence compared to placebo, a finding that is directly relevant to adults over 50 who have had polyps found on colonoscopy. Astaxanthin, the carotenoid pigment that gives wild salmon its characteristic pink-red color, is one of the most potent antioxidants in nature, with an oxygen radical absorbance capacity approximately 550 times greater than vitamin E. In cancer biology specifically, astaxanthin has demonstrated the ability to inhibit the formation of new capillaries around tumor spheroids in laboratory models, and to reduce the expression of hypoxia-inducible factor 1 alpha, the master regulator of the tumor's angiogenic response to hypoxia, in multiple cancer cell lines. 3 servings of wild salmon per week, each approximately 4 to 6 ounces, provides the combination of EPA, DHA, and astaxanthin at doses associated with measurable anti-inflammatory and anti-cancer effects in clinical populations. Number 3 is pomegranate. And I place pomegranate here because the clinical evidence for its anti-angiogenic activity in humans is among the most directly validated of any food I could place on this list. A randomized controlled trial published in the Proceedings of the National Academy of Sciences found that pomegranate juice consumed daily for 1 year produced not only reductions in PSA velocity in men with prostate cancer, a measure of how quickly PSA levels are rising that is a proxy for tumor growth rate, but measurable inhibition of the angiogenic signaling in prostate tissue. The control group showed the expected increase in PSA velocity. The pomegranate group showed a dramatic slowing. This is not a surrogate marker. This is evidence of reduced tumor growth activity in a living human being from consuming a dietary intervention. The mechanism involves punicalagins and punicalins, polyphenols found almost exclusively in pomegranate, which are converted by gut bacteria to urolithin A. Urolithin A has demonstrated extraordinarily potent inhibition of VEGF signaling, direct pro-apoptotic activity in several cancer cell lines including breast, colon, and prostate, and enhancement of natural killer cell cytotoxicity against tumor cells in multiple laboratory models. Pomegranate also contains ellagic acid, which has demonstrated in research published in the journal Cancer Prevention Research the ability to inhibit the formation of heterocyclic amines, the carcinogenic compounds produced when meat is cooked at high temperatures, and to induce what researchers call phase 2 detoxification enzymes in the liver that neutralize carcinogens before they can damage DNA. This dual activity, inhibiting carcinogen formation and accelerating carcinogen detoxification, makes pomegranate especially relevant for adults over 50 who consume any amount of cooked meat. Half a cup of pomegranate seeds or 4 to 6 ounces of pure pomegranate juice without added sugar daily provides the polyphenol dose associated with the anti-angiogenic effects I described. Number 2 is broccoli, but not in the way most people have been eating it, and not for the reason most people think. And I want to spend more time here than on any other food except the number 1, because the preparation error with broccoli is one of the most consequential mistakes in anti-cancer nutrition that I have seen in my clinical experience. The compound that makes broccoli extraordinarily powerful as an anti-cancer food is sulforaphane, an isothiocyanate that activates the NRF2 transcription factor, the master switch of the body's endogenous antioxidant and detoxification defense systems. When NRF2 is activated by sulforaphane, it triggers the expression of more than 200 protective genes, including the glutathione synthesis genes that produce the body's primary intracellular antioxidant, the heme oxygenase 1 gene that protects against oxidative damage, and several genes that directly inhibit tumor necrosis factor alpha and interleukin 6, two of the primary inflammatory mediators that create the tumor-permissive microenvironment. Sulforaphane also has direct anti-cancer mechanisms that are independent of NRF2. It inhibits histone deacetylases, enzymes that silence tumor suppressor genes, effectively reactivating the genes that cancer cells have shut down to escape cell cycle control. It induces apoptosis in cancer stem cells, which are the small subpopulation of cells within tumors that are responsible for metastasis and for the regrowth of tumors after treatment. And in a study published in Cancer Prevention Research that enrolled smokers with high cancer risk, sulforaphane supplementation, equivalent to 2 cups of broccoli, daily reduced urinary carcinogen metabolites by 87% compared to placebo. 87% reduction in the carcinogens reaching the DNA of the high-risk cells in their airways. But here's the preparation error I mentioned. Sulforaphane does not exist preformed in broccoli. It is produced when an enzyme called myrosinase comes in contact with its precursor, a compound called glucosinolate. This enzymatic reaction happens when broccoli cells are damaged, which is when you cut or chew it. The problem is that myrosinase is exquisitely sensitive to heat. When you cook broccoli at high temperatures, boiling, steaming excessively, or microwaving on high, you destroy the myrosinase before it can convert the glucosinolate into sulforaphane. The glucosinolate remains as glucosinolate and the anti-cancer activity is essentially eliminated. The 2 solutions are these. First, eat broccoli raw or lightly blanched, meaning submerging it in boiling water for no more than 90 seconds and immediately transferring it to ice water to stop cooking. The rapid heat exposure is insufficient to destroy the myrosinase. Second, and this is the approach I use personally, cut the broccoli and let it rest for 40 minutes before any cooking. This rest period allows the myrosinase to fully complete the glucosinolate to sulforaphane conversion while the enzyme is still active. After 40 minutes, you can cook the broccoli with moderate heat and retain most of the sulforaphane that has already been formed. Combine broccoli with mustard seeds, which contain their own myrosinase that can compensate for heat-inactivated broccoli myrosinase, and you have maximized the sulforaphane yield regardless of cooking method. 2 cups of broccoli prepared correctly 3 to 4 times per week is the dietary dose that the research associates with measurable reductions in cancer biomarkers, and the same principle applies to other cruciferous vegetables. Cauliflower, Brussels sprouts, kale, and arugula all contain glucosinolates that produce anti-cancer isothiocyanates when the myrosinase reaction occurs correctly. And now we arrive at number 1, the food that the MD Anderson researchers ranked as the most powerful anti-angiogenic food in their 9-year study. The food that I promised would surprise you. The food that many adults over 50 have been told to limit or avoid due to concerns about fat and calories. It is avocado. Specifically, the combination of avocado's unique fatty acid profile, its sterile content, its remarkably concentrated antioxidant suite, and its specific effect on the metabolic pathways that cancer cells depend on for their energy supply. Let me explain each component in the detail it deserves, because the mechanisms here are genuinely extraordinary. The first mechanism is lutein. Avocado is one of the richest sources of lutein per gram of any food, with concentrations comparable to dark leafy greens per calorie. Most discussions of lutein focus on eye health, and I have addressed that at length in other videos on this channel. But lutein's anti-cancer activity is separate from and equally compelling to its ocular protective effects. A study published in the Journal of Nutritional Biochemistry found that lutein-induced apoptosis in breast cancer cell lines inhibited the proliferation of colon cancer cells and reduced the adhesion and invasion capacity of lung cancer cells in laboratory models, all through mechanisms involving the activation of caspase-dependent apoptotic pathways and the inhibition of the NF-κB inflammatory signaling that cancer cells use to resist programmed cell death. The second mechanism, and this is the one that most directly connects to the cancer-starving theme of this video, is avocado's unique ability to target leukemia stem cells through a compound called avocatin B. Avocatin B is a lipid found exclusively in avocados, and it has demonstrated in research published in the journal Cancer Research the ability to selectively induce apoptosis in acute myeloid leukemia stem cells while leaving normal stem cells unharmed. The mechanism is through the inhibition of fatty acid oxidation in the mitochondria of leukemia stem cells specifically. Leukemia stem cells, unlike their normal counterparts, depend on fatty acid oxidation as their primary energy source rather than glucose. Avocatin B disrupts this specific metabolic dependency, literally starving the leukemia stem cells of their fuel, while normal cells, which have metabolic flexibility and do not depend exclusively on fatty acid oxidation, are unaffected. This is exactly the kind of metabolic disruption that the MD Anderson researchers were referring to when they used the term natural metabolic disruptor for tumor cells, a compound that selectively targets a metabolic vulnerability of cancer cells that normal cells do not share. The third mechanism is avocado's extraordinary content of glutathione, the body's primary intracellular antioxidant. Avocado is one of the highest natural sources of glutathione available in any food, with concentrations significantly higher than most vegetables and fruits. Glutathione's relevance to cancer prevention is not simply as an antioxidant in the generic sense. It is the primary substrate for the phase 2 detoxification enzymes that neutralize carcinogens in the liver and in the mucosal surfaces of the digestive tract. High intracellular glutathione is one of the most consistent characteristics of tissues that resist carcinogen-induced DNA damage, and high dietary glutathione from avocado and other sources has been associated in population studies with reduced cancer incidence across multiple tumor types. The fourth mechanism is beta-sitosterol, a plant sterol that avocado contains in concentrations that are among the highest of any food. Beta-sitosterol has demonstrated in multiple peer-reviewed studies direct anti-proliferative activity against prostate cancer cells, breast cancer cells, and colon cancer cells through mechanisms that include the inhibition of the cell cycle at the G1S transition, the suppression of NF-κB signaling, and the induction of apoptosis through the mitochondrial pathway. A study published in Anti-Cancer Research found that beta-sitosterol from avocado was among the most potent naturally occurring inhibitors of prostate cancer cell proliferation tested in that study. The fifth mechanism is the one that connects avocado directly to the whole grain toast I told you to stop eating at the beginning of this video. Avocado is extraordinarily low in digestible carbohydrates and high in healthy monounsaturated fat and fiber. When you replace the high glycemic load of whole grain toast with avocado, you are doing something that is directly anti-cancer in its metabolic effect. Cancer cells, as I described in the context of the Warburg effect, preferentially and in many cases almost exclusively use glucose as their energy source. They express high levels of glucose transporters and glycolytic enzymes that make them aggressively efficient at capturing circulating glucose. In a high glycemic dietary environment, with repeatedly elevated blood glucose and insulin levels after every meal, this metabolic preference of cancer cells is continuously being fed. In a low glycemic dietary environment, where blood glucose is stable and insulin remains relatively low, the preferential metabolic fuel of cancer cells is less available. Avocado, with its minimal glycemic impact combined with its satiating fat and fiber content, creates exactly the low glycemic metabolic environment that is less hospitable to the energy supply of cancer cells. This is not about a ketogenic diet or any extreme dietary approach. It is about the metabolic consequences of replacing a high glycemic breakfast staple with a food that provides equivalent satiety with dramatically lower glucose and insulin impact. A study published in the American Journal of Clinical Nutrition that specifically examined the relationship between dietary glycemic load and cancer recurrence in breast cancer survivors found that women in the highest quartile of dietary glycemic load had a significantly higher risk of cancer recurrence compared to those in the lowest quartile. The difference was driven primarily by the frequency and magnitude of post-meal glucose and insulin spikes, which are directly addressed by the substitution of avocado for high glycemic carbohydrates at breakfast. Half an avocado daily, consumed as part of a meal with other protein and healthy fat, provides the lutein, avocatin B, glutathione, and beta-sitosterol in doses that the research associates with meaningful anti-cancer effects. And it does this while creating the low glycemic metabolic environment that is directly hostile to the preferential energy metabolism of cancer cells. Now let me bring all 5 foods together into the framework that makes their combination more powerful than any individual food alone. The 5 foods I have described today each engage different mechanisms of cancer prevention. Green tea and matcha inhibit angiogenesis and enhance immune surveillance. Wild salmon shifts the lipid balance away from pro-cancer inflammatory signaling. Pomegranate provides unique polyphenols that inhibit VEGF and enhance phase 2 detoxification. Broccoli activates the NRF2 pathway that upregulates the body's endogenous anti-cancer defenses, and avocado provides unique lipids that selectively target cancer stem cell metabolism while creating a systemic low glycemic environment that is metabolically hostile to cancer cells. No single one of these foods works in isolation to prevent cancer, but their consistent, regular incorporation into a dietary pattern that also eliminates the primary dietary drivers of cancer risk, the refined sugars, processed meats, refined vegetable oils, and highly processed foods that promote the inflammatory microenvironment in which cancer initiates and progresses, creates a cumulative biological environment that the research identifies as dramatically more protective than any single intervention. The MD Anderson study found a 47% reduction in cancer recurrence with consistent consumption of anti-angiogenic foods. The Harvard School of Public Health cohort study that I have discussed in previous videos found a 61% reduction in cancer risk from consistent healthy lifestyle behaviors including dietary pattern. These are not marginal effects, they are biological signals of the magnitude that drug trials struggle to achieve, coming from food choices that are accessible, affordable, and immediately actionable. I want to close by addressing the question that I know many of you are asking internally, which is whether it is too late to start making these dietary changes if you are already in your 60s, 70s, or beyond. And my answer, based on the biology of carcinogenesis, is unequivocally no. The process by which a normal cell becomes a cancer cell takes 10 to 20 years on average. It passes through multiple stages, each of which has biological checkpoints that can be influenced by the dietary environment. The NRF2 pathway that sulforaphane activates operates in cells of any age. The VEGF inhibition that EGCG and pomegranate provide is relevant at any stage before a tumor becomes angiogenic. The natural killer cell enhancement from omega-3 fatty acids and the anti-cancer stem cell activity of avocatin B are not age-limited mechanisms. They work in the biology of a 70-year-old just as they work in the biology of a 50-year-old. What is different after 60 is not whether these dietary interventions work. It is that the biological systems they support have been declining for longer, meaning the cumulative deficit to address is greater and the urgency of starting is higher. The best time to begin eating this way was 20 years ago. The second best time is this week. I am Dr. William Lee. If this video gave you something valuable today, please share it with someone over 50 in your life who is thinking about cancer prevention or who is navigating a cancer recovery. Subscribe to this channel for more evidence-based oncology and nutrition content. And leave me a comment below. Tell me your age. Tell me whether cancer has touched your life or the life of someone you love. And tell me which of these 5 foods you are going to add to your diet this week. Your body has the biological capacity to resist cancer. Feed it what it needs to exercise that capacity.